This is a best-in-show champion, the absolute peak of canine genetics. However, this animal is a replica, a multi-million-dollar carbon copy, a $50,000 carbon copy, gestated inside a sterile facility. The elite are no longer breeding their champions, they are printing them. The process is called somatic cell nuclear transfer. It begins with a single tissue biopsy, harvested from a deceased winner. Technicians culture the cells for 2 weeks, multiplying the DNA of a ghost. They extract a donor egg from a rented lab dog. A clone cannot be grown in a glass jar. It requires a living incubator. They hollow it out entirely, stripping away its original identity. The champion's nucleus is injected inside. An electrical shock forces the cell to divide. But the failure rate is catastrophic. Animal welfare activists track this hidden population. They confirm a grim mathematical reality. Most pregnancies end in termination or severe biological glitches. The world's first cloned dog required 1,095 embryos, 123 surrogate mothers. The pregnancy success rate hovers at 40% to produce one surviving animal. The industry has streamlined the sacrifice since then, but it remains an industrial pipeline, operating completely in the open. Clients expect an exact replica. They assume the ribbon comes encoded in the DNA. Genetics can copy the physical frame, the exact curl of the coat, the precise structure of the jaw. They do not copy the mind. They do not copy the 10,000 hours of training. A clone begins with zero memory. It is a genetic stranger wearing a familiar face. Yet the market for these animals is exploding, driven by vanity and the desperate refusal to let a winner die. To understand what happens inside these facilities, you must look past the pristine reception areas. The global demand is staggering. It began with military and police contracts, breeding the top 1% of working dogs, generating millions of dollars in annual revenue. But the market quickly shifted to the ultra-wealthy. The Crown Prince of Dubai was an early client. A British couple spent a fortune to clone their boxer. The trend normalized rapidly among American elites. Tom Brady cloned his family dog. Barbra Streisand cloned hers, naming the two identical puppies Miss Violet and Miss Scarlet. This celebrity client list provides a shield of legitimacy, but the true engine of this industry is the show ring. Natural breeding is a biological lottery. 2 champion parents do not guarantee a champion puppy. The elite are tired of gambling. A best-in-show winner is a high-yield financial asset. Cloning removes the unpredictability of natural reproduction, or so the marketing brochures claim. Handlers exchange 6-figure checks for genetic consistency, buying a blueprint they already know works. They believe they are purchasing a finished product, an animal mathematically engineered to win. Every industry has an architect, someone who built the machine before anyone else arrived. In the early 2000s, a single government-funded research unit held every patent that mattered. A leading international science journal published 2 landmark papers from this team, 2004, 2005. Claiming breakthroughs the entire global biomedical community had been chasing for years. The national government awarded the research unit over $1.5 million in funding. The scientist leading the work was declared a national hero. He had achieved something else first. According to a peer-reviewed biology journal, his team had successfully produced the world's first cloned dog, a genuine, independently verified biological result. The dog cloning was real. The stem cell research was not. The state-run university launched a formal internal investigation. According to the European Union's official science reporting, the investigation concluded that both major research claims had been entirely fabricated. The journal formally retracted both published papers. The scientific community called it one of the most consequential frauds in modern biology. The scientist was dismissed from the institution, then prosecuted by the state. According to multiple international press reports, a district court found him guilty of embezzling public research funds and illegally acquiring biological material. He received a suspended sentence. According to International Wire reporting, the country's highest court upheld that conviction in 2014. The case was closed. He did not stop cloning dogs. According to press reporting in South Korea, he launched a new private research foundation in 2006. The patents over the dog cloning process were contested in civil court. According to those same court records, he ultimately retained the right to operate commercially. The commercial pet cloning industry did not emerge from clean academic research. It was built on infrastructure created during a period of documented scientific fraud. According to financial press reporting, clients were paying $100,000 per clone, with no legal requirement to disclose that history to buyers. The regulatory vacuum was not accidental. The founding scandal had already demonstrated exactly what oversight this industry would face. None. The operation that wealthy clients now pay $50,000 to access was not born from legitimate science. It was salvaged from the collapse of it. And then it was scaled into a global market. But the founder's history with falsified data carried a warning the industry chose to ignore. You cannot falsify what happens inside a cell. Biology always introduces flaws, and those flaws are deliberately minimized in the sales pitch. The industry's marketing material sells certainty. The largest commercial lab makes a specific claim online. They state clones often share the original's intelligence and often share the same temperament. A cloned dog carries identical nuclear DNA, but it does not inherit the exact physical markings. Coat color and spot patterns differ between identical clones. These are influenced by epigenetics during embryonic development. The clone looks similar, but it is not a photocopy, and the cosmetic differences are merely the surface. Documented abnormalities plague the cloning pipeline. The glossy brochures omit the biological casualties. Offspring are frequently born with severe cardiovascular defects. Immune systems fail to develop properly. Many suffer from large offspring syndrome, an abnormal birth size that threatens both puppy and surrogate. Some are born with enlarged tongues or cleft palates. The early mortality rate is significantly elevated. The biological degradation compounds over time. Serial cloning tests the limits of genetic stability. In a documented case, a clone of a clone was born. an attempt to stretch a champion's bloodline a third time. It died on the day of birth, killed by massive liver and gallbladder anomalies. The genetic code breaks down with repeated copying. The industry knows the product is biologically fragile. Yet clients ignore the biological warnings. They focus entirely on the promise of the familiar face. They purchase the physical shape of their champion, assuming the personality will somehow follow. It is a fundamental misunderstanding of biology, setting up a devastating psychological collision. When the buyer finally meets their $50,000 stranger, the psychological toll is immediate. The industry sells the continuation of a bond, but the client receives a blank slate, a biological reset to zero. The origin of performance cloning proves this perfectly. In 2009, a lab cloned a hero. The original dog pulled the last survivor from the World Trade Center. His handler commissioned the procedure. 5 exact genetic replicas were produced. None of them ever searched a single pile of rubble. They looked exactly like the hero. They possessed none of his experience. Despite this known reality, the market is expanding rapidly, valued between $145 million and $319 million annually. The true scale remains deliberately opaque. There are no international oversight committees, no laws governing the surrogate networks, and zero consumer protections for the buyers. A champion is the product of 10,000 hours of conditioning, built on desensitization to stadium lights and crowds. It requires a profound, specific trust between two species, a relationship forged over years. You cannot clone a relationship. You can only rebuild it from zero. Show dogs are merely the prototype. The pet cloning market is funding a much larger ambition. It shares the exact same corporate infrastructure, the exact same cellular preservation pipeline as the companies attempting to resurrect extinct species. The technology perfected on a childhood pet is being scaled to reverse history entirely. In late 2025, one of the largest pet cloning operations in the world was acquired. The buyer had one declared goal: to bring back the woolly mammoth. The technique is identical. The pipeline already exists. The target date is 2028. The limits of this technology are already being tested. One lab holds the current record. They produced 50 living copies of a single horse. Dozens of clients have ordered clones in the 20s. The technology is no longer limited to grieving pet owners. Governments are already placing institutional orders. Military programs are cloning their highest performing working animals. The logic is simple. If the genetics produced one elite animal, replicate the genetics. But the physical toll of mass production is severe. Scientists have already cloned the original clones, stretching a single lifespan across 3 separate bodies, proving that genetic material decays with every forced reboot. The price of a clone has already dropped 40% in a decade. Industry analysts project it will fall below $10,000 within 5 years. At that price point, this is no longer a luxury. It becomes a consumer decision. The same dog for every generation of your family. The same face at every birthday, every Christmas, every goodbye. Death becomes optional, which leaves only one question. What exactly is the buyer taking home? Natural selection built the domestic dog over 15,000 years, every generation producing variation, every variation tested against survival. Cloning removes that process entirely. A copied animal did not survive because it was the fittest. It survived because someone signed a check. According to a study published by the National Institutes of Health, over 1,000 cloned dogs have now been documented. The data shows consistent biological anomalies absent in natural reproduction, including cases of sex reversal across multiple breeds. The genetic divergence compounds with every copied generation. The industry is narrowing the gene pool it claims to be preserving. And the dog is only the beginning. According to UNESCO, more than 50 countries have now passed laws specifically banning the application of this technique to human beings. The technology is identical. The legislation exists precisely because the capability does too. The same somatic cell nuclear transfer used on a show poodle in 2005 is the same procedure those laws were written to prevent. The only difference between a cloned dog and a cloned human is a law that does not exist in every country. Look back at the archetype. A 3-year-old champion winning the highest title in the sport. Over 2,500 competitors entered that ring. That specific victory belongs to the original animal alone. You cannot duplicate the ribbon. The buyers know this eventually. The money does not purchase the champion. It purchases the shape of the dog that used to be one, a biological monument to an animal they refuse to let go. The industry sells a ghost. The cells do not carry the years of training. They do not carry the patience of the handler or the 10,000 hours of conditioning. They carry the genome. The industry has decided that is close enough. But nature does not negotiate with grief. You can replicate the pedigree. You can print the perfect physical shell. Yet when the stadium goes dark and the buyer is finally left alone with their $50,000 stranger, the illusion breaks. Did they bring their champion back? Or did they just pay a fortune to prove it is actually gone?